Hello guys, so welcome back. Today we shall be discussing another most important bacteria. All of you know that is your Bordetella pertussis, right? So when I tell pertussis, right, what do you mean by that? So I know pertussis, what do you mean by that is it is something related to the cough, right? So Bordetella pertussis is the bacteria that is responsible for causing whooping cough. So what is whooping cough? Why does it cause whooping cough, right? So why is it not a normal cough? We shall be discussing all the mechanisms in much detail. But before that, we shall discuss, as it is a microbiology session, we shall be discussing the general properties of the bacteria. We shall be discussing the different types of mechanisms, right? So we shall start over it. So first of all, let us discuss about the general characteristics of the bacteria. So let us discuss about the general characteristics of the bacteria. Right. So coming to the general characteristics of the bacteria, what are the important things you need to keep in mind? So the first important thing you need to keep in mind is that whether this is a gram positive or a gram negative bacteria. So obviously this is a gram negative bacteria. So what do I mean by gram positive bacteria? Those bacteria which are having a thick peptidoglycan layer is called as gram positive bacteria, and those bacteria which are having thin peptidoglycan layer is called as gram negative bacteria. So this bacteria is gonna have thin peptidoglycan layer, right? So this bacteria is going to have thin peptidoglycan layer. So because of this thin peptidoglycan layer, you call this bacteria as gram negative bacteria, right? So what type of bacteria is this, guys? What type of bacteria is this? Is this a cocci or a bacilli? It is a gram negative cocobacilli. Gram negative cocobacilli bacteria, right? Next, third important thing which we need to discuss here, and it is very important, that whether this bacteria can move or not. So this bacteria is not having any kind of flagella. So if this bacteria is not having any kind of flagella, I call it as a non-flagellated bacteria. So obviously that bacteria which does not have any kind of flagella cannot move. So it is a non-motile. So it is a non-flagellated bacteria. So as a result of this bacteria is a non-flagellated, so it can be a non-motile bacteria. So this is a non-motile bacteria. Next important thing which you need to keep in mind is that this bacteria, when you look at the bacteria, so some of the strains of this bacteria are having a slime layer surrounding it. For example, let us say this is a bacteria over here. This is the bacteria, right? Now surrounding this bacteria there is a layer of slime. So this is a bacteria. Now surrounding this bacteria there is a layer of slime. You see this layer now which I am drawing here. So this layer which I have drawn here, this one is called as your slime layer, right? So this is called as the slime layer of the bacteria. So what is this slime layer? What is the other name for this slime layer, guys? The other name for this slime layer is also called as biofilm. So what do I mean by that is that, for example, bacteria is having a sticky slime layer, right? So why is it so important? Because with the help of that sticky slime layer, it will very easily stick to your skin surface. Okay, it will very easily stick to your skin surface. Or for example, let us say that you are injecting some needles into the vein, right? So you are injecting the catheter into the vein. So it will very easily, with the help of this slime layer, it will very easily stick to the needle, right? So this slime layer is also called as biofilm. Whereas some bacteria will have another important thing surrounding this, like this. Look here. Surrounding this, some of the bacteria have some more important things, and this thing you call it as a capsule. So can I call that this bacteria is having a slime layer and some of the bacteria are having a capsule around it? So what do you call? You call them as encapsulated bacteria. These are called as encapsulated bacteria. What type of bacteria you call this, guys? You call this type of bacteria as encapsulated bacteria. right? So slime layer, biofilm, encapsulated bacteria, non-flagellated, non-mobile, gram-negative coccobacilli. Next, coming to the real properties. What are the real properties of this bacteria? This bacteria is catalase positive, right? So this bacteria is catalase positive. Now, what is your concept of catalase positivity? What do you mean by catalase positive? Catalase positive means that bacteria which has an ability to release an enzyme called catalase, you call it as catalase positive organisms. For example, let me draw it here. For example, this is a bacteria over here, right? And this bacteria has a capacity to release an enzyme, and that enzyme you call it as catalase. That enzyme you called as catalase. And what is the function of catalase, guys? What does catalase do is that catalase will basically convert H2O2. What is H2O2? H2O2 is hydrogen peroxide. So catalase converts H2O2 into water plus half mole of oxygen. Okay. So water and half mole of oxygen together you call it as a foam. For example, let us say this is your blood vessel, right? So I know most of you know what I am trying to teach, but for those of you who haven't attended any of my lectures and this is your first lecture, so you have to know what is the importance of catalase. Okay. So let me also draw some important things, like these are the endothelial cells. All of you know your blood vessels are comprised of endothelial cells like this, right? Now, for example, let us say that you have got some superficial cut wounds, right? Some superficial bruises and all. So what do you add? You add hydrogen peroxide. When you add hydrogen peroxide, you see that white color foam coming out of that. That is nothing but water and oxygen, right? So always and always add hydrogen peroxide only to the superficial wounds. Never ever add hydrogen peroxide to deep cut wounds. For example, let us say my radial artery, my ulnar artery, my brachial artery, my femoral artery, if they are cut, right, never ever add hydrogen peroxide there. Let us say this is a radial artery or whatever it is. This is some artery that is situated deep inside your body. And let us also say that there is a damage to this artery over here. Now, when there is a damage to this, obviously all of you know the blood oozes out like this, right? So, at this damaged region, you add hydrogen peroxide. Now, when you add hydrogen peroxide, what is happening is within these endothelial cells, right? If you very closely look, guys, from the ruptured endothelial cells or from the broken, damaged endothelial cells, an enzyme is released, right? So, that enzyme is your catalase. Obviously, it is also released from bacteria. So, what happens with this catalase now? What happens is that the moment you add hydrogen peroxide H2O2, right? Hydrogen peroxide H2O2 gets converted into water plus half mole of oxygen. It means within this blood vessel, a lot of oxygen molecules are found. So, all these oxygen molecules within this blood vessel, what do they cause? Yes, what do they cause is that they cause air embolism. So, this is one of the very important thing one has to remember while you are in your clinic. Okay. So, this is an importance of catalase. So, this bacteria releases a catalase enzyme. So, this is called as a catalase positive organisms. Not only catalase, guys, it also releases another enzyme that is called as cytochrome oxidase. So, if it is releasing cytochrome oxidase, can I call it as oxidase positive? Yes. So, this bacteria is also oxidase positive. Right. Next important thing which you need to know is that this bacteria, see, most of the bacteria, for example, Klebsiella pneumonia, right? So, those bacteria, if you add citrate to them, they utilize that citrate. They think citrate is a source of energy and they utilize that citrate. But if I am adding citrate to this bacteria does not utilize citrate. So, can I call this bacteria as citrate negative? Yes, this bacteria is citrate negative bacteria. So, these are some of the important things which you have to know, that this bacteria has got a thick peptidoglycan layer. So, because of the thick peptidoglycan layer, I am calling it as a gram-negative bacteria. And this bacteria is non-flagellated, and all of you have to keep this thing in mind, that this bacteria has got what? Biofilm, right? And all of you will definitely know that it is catalase positive, it is oxidase positive because it releases what? Cytochrome oxidase, and then it is citrate negative because it does not utilize citrate as a source of energy. Right. So, how does this bacteria basically enter into your body? Right. How does this bacteria enter and how is this causing a disease? Okay. So, this bacteria basically enters through your upper respiratory tract. Okay. So, let me draw a picture and explain you the exact mechanism of how this bacteria is pathogenic to you. So, let us discuss the pathogenesis. Now, we are discussing the pathogenesis. Pathogenesis part. So, what is the important thing that you have to remember while discussing the pathogenesis? So, let me draw the lateral surface of the face, guys, the lateral surface of the face, and let me explain it to you. For example, this is your lateral surface of the face. Right. So, for example, this is your lateral surface of the face. Now, in this lateral surface of the face, here, what do you have here? Yes, here you have your nasopharynx like this. This is your nasopharynx. Next, here comes your oropharynx. So, this one which I am drawing right now is your oropharynx. So, your nasopharynx, oropharynx together, they join together and form your laryngopharynx. Right? So, what is this, guys? Tell me. What is this thing? This is your nasopharynx. This is your nasopharynx. And this one is your oropharynx. So, nasopharynx, oropharynx together they form the laryngopharynx. So, this bacteria either can enter through oropharyngeal way or through nasopharyngeal way. So, all of you know that you have got some very specialized epithelial cells here, isn't it? You have got some very specialized epithelial cells like this. Look here. These are some of the specialized epithelial cells. Now, these specialized epithelial cells, these are called as pseudostratified ciliated columnar epithelium. This will be very important for you in histology. So, let me tell it to you right now so that you don't confuse later on. This is pseudostratified ciliated columnar epithelium, right? So, what type of epithelium is this? This is pseudostratified ciliated columnar epithelium, right? It means it has to have cilia, right? So, there are also cilia that are located. Let me use another color. So, there are also abundant amount of cilia that are located on this, right? So, what are you trying to teach us? What am I trying to teach you is that, guys, in between these pseudostratified ciliated columnar epithelial cells, there are some specialized cells. Let us say these are called as black cells. Okay, I mean actually they are not called as black cells, but as far as my lecture is concerned, I am telling these as black cells. So, these black cells are called as goblet cells. And what is these black cells are called? They are called as goblet cells. So, what is the function of goblet cells, guys? Goblet cells release what? They release mucus. Goblet cells release mucus. So, in this patient right now, what is happening? Now, I bring up a bacteria in the nasopharynx. This is your pertussis bacteria, right? Now, this bacteria all the way enters into your nasopharynx. Now, the moment this bacteria enters into your nasopharynx, what is going to happen? What is going to happen is that this bacteria will not stay there. It will start to move. It will start to giggle. So, when this bacteria is in your oropharynx or the nasopharynx, it will irritate your cilia. It will irritate your epithelium. It will even irritate your goblet cells. So, when it is irritating your goblet cells and when it is irritating all of this stuff, then what is happening in this patient, guys? Can anyone tell me what is happening? What is happening is there will be excess amount of mucus secretion. For example, if I take a feather and irritate your nasal region, obviously you sneeze out. So, why? What is happening? Because you are irritating the epithelium over there, right? So, in the same way, this bacteria in the nasopharynx and the oropharynx, it starts irritating that. When it starts irritating that, there will be increased mucus secretion. Increased mucus secretion. And who is secreting this mucus? The goblet cells, obviously. So, increased mucus secretion. And whenever there is increased mucus secretion, what is going to happen? There are cilia, a lot of mucus is flowing from both the sides, and there is bacteria in the center. So obviously you will cough, right? You will cough with the sputum. So that is a thing why sometimes whenever you are going outside exposed to some pollution and all, you cough. Why? Because those irritants are entering into your respiratory tract, that is causing increased mucus secretion, and increased mucus secretion will allow you to cough out. So when you cough out, what is happening is that there is a lot of mucus now. There is a lot of mucus here. All this mucus along with the bacteria, it is expectorated outside. It is expectorated outside. This happens with every bacteria. This happens with every irritant, every microorganisms that is entering into your body. But with Bordetella pertussis, the scene is completely different. The mechanism is completely different. Why it is different? Why it is different is that, look what I'm going to draw is, I'm just going to take this part, guys, concentrate here. I'm just going to take this part and draw it down and explain you what is exactly happening. Okay, so look here now. For example, so let me draw the pharyngeal wall here. So this is the wall. This is the wall, right? Now all of you know, you have got your cells that are here, cells that are located here. What are these cells? See, these are the pseudostratified ciliated columnar epithelial cells, right? So these cells are pseudostratified ciliated columnar epithelial cells. And these cells, obviously all of you know that they have got cilia on its surface, right? So let me draw these cilia little bigger so that it will be better for me to explain. So let me draw this cilia little bigger over here. Now concentrate here what is happening. The bacteria is obviously, see, this entire part, this is called as your lumen, isn't it? This entire part is called as your lumen. So right now that bacteria is exactly here in the lumen. This bacteria is exactly right now here in the lumen, right? Now what is this bacteria going to do? First thing what will happen is that this bacteria, if it has to enter into my body, enter into my bloodstream, this bacteria should be attached to those cilia, attached to those cells very firmly. So what will happen is that this bacteria has got some elongated chains like this, right? You see these elongated chains? So what am I doing with these elongated chains? With the help of these elongated chains, this bacteria is holding the cilia like this on both the sides, right? So these elongated chains are called as filamentous process, right? So these elongated chains are called as filamentous process. The exact name of this is filamentous hemagglutinin. This is called as filamentous hemagglutinin, right? Not only that, guys, not only filamentous hemagglutinin, there are also some pinkish molecules like this. You see, bacteria is also having some pinkish molecules, and these pinkish molecules are called as pertactin. These pinkish molecules are called as pertactin. Can you please explain us once again what are you trying to tell? What am I telling is that the remaining bacteria, they don't have this type of filamentous hemagglutinin or pertactin molecules. So basically when the bacteria comes there, it will irritate the globulate cells, and the globulate cells will release mucus. Mucus along with the bacteria, it is expectorated outside. But for this pertussis is having your specialized filamentous hemagglutinin. Not only that, it is also having pertactin. So because of these two molecules, it is very firmly adhering to the host cell. If you ask me exactly where in the host cell, it is very firmly adhering to the cilia of the host cell. Okay, fine. So when it is adherent to the cilia of the host cell, even if the cilia are moving, the bacteria doesn't move because they are very firmly attached. Now this bacteria releases a special toxin, guys, right? It releases a special toxin. Look here now. This bacteria is right now releasing a special toxin. These blue dots which I am drawing here, this is the special toxin that is released by this bacteria. You know what is the name of this toxin? Yes, this toxin is called as tracheal cytotoxin. The name of this toxin is tracheal cytotoxin. Why is tracheal cytotoxin is released? The reason is that this tracheal cytotoxin will paralyze all the cilia. It will damage the structure, the ultrastructure of the cilia, and it will paralyze all the cilia. So when it is paralyzing all the cilia, previously cilia were wavy like this. So now you know what will be the shape of the cilia? The cilia will become like this, straight, completely straight. Now the cilia will become completely straight in this way. The cilia are no more moving. They are completely straight. So when there is no ciliary movement, obviously you cannot push the bacteria up and expectorate outside. So first thing, what did the bacteria do? The first thing the bacteria did is that, first of all, it anchored itself. Second thing what it did is that it released a toxin called as tracheal cytotoxin. And this tracheal cytotoxin, right, it paralyzed all the cilia over there. Right, this tracheal cytotoxin paralyzed all the cilia over there. So when it paralyzed all the cilia over there, what is happening? So let me write it down, that this tracheal cytotoxin paralyzed the cilia. Right. So when it paralyzed the cilia, what is going to happen? Now slowly this bacteria enters into the epithelial cell. Right. So how is it entering into the epithelial cell? It will enter into the epithelial cell by a process called as endocytosis. Look here. Now slowly this bacteria, by a process called as endocytosis, this bacteria enters inside the cell. Can you tell me the exact structure of the bacteria? The exact structure of the bacteria, even I don't know. Obviously I told you it is coccobacilli, right? But within this bacteria, there are two subunits, guys. There are two subunits within this bacteria. Let me write it down. So one is called as the A subunit and another one is called as the B subunit. So A and B subunit together form this bacteria. Okay. So this A and B subunit of this bacteria, finally it entered through a process called endocytosis. Now this bacteria is right now inside your epithelial cell. Right. So it has got two toxins. One is called A subunit and B subunit. Now out of this A and B subunit, through some molecular processes, what happens is that the B subunit goes away. So when the B subunit goes away, what is left free? Yes, what is left free? Obviously the A subunit. So this A subunit is the one which is very toxic. Okay. Similar mechanism I have explained in cholera. Even in the cholera, there are A and B subunits, out of which the B subunit goes away and A subunit comes into the cytoplasm, and that is very toxic. So here also the A subunit is very toxic. You know what will happen with this A subunit? Look here. Let us go to the picture back. Now surrounding your wall, obviously all of you know that you have got your blood vessel like this. Definitely you will have your blood vessels. So I am going to draw one blood vessel of like that in this way here. So there is a blood vessel that is supplying to your oropharynx or your nasopharynx. So this is a nasopharyngeal artery or an oropharyngeal artery, whatever it is. Now this A subunit slowly enters into your blood vascular system. After entering into the blood vascular system, what is it going to do? What is it going to do is that this A subunit will release a toxin, right? And that toxin, look here, it is going to release these small green dots which I am showing you here. These small green dots are nothing but a toxin, and this is called as pertussis toxin. Okay. So this toxin which is released by this bacteria over here, as you can see. So this toxin which is released by this bacteria, this is called as pertussis toxin. So can you tell me some important things about this pertussis toxin? What this pertussis toxin is going to do? What this pertussis toxin will do is that, point number one, this A subunit is releasing this pertussis toxin, and right now that is in the blood vascular system. Now this toxin will call all the lymphocytes in the body. So all the lymphocytes will come and gather there. Look here now. All the different types of lymphocytes are coming and gathering here. So what will happen when all the lymphocytes will gather? So there will be increased lymphocytosis in the patient. Increased lymphocytosis. Not only that, can you tell me the second thing? The second thing is that, guys, what is the basic function of histamine? Histamine is responsible for vasodilation. All of you know that, right? Then histamine is normally present in the blood. Obviously, it is heavily loaded in the mast cells, but some amount of histamine is also present in your blood vascular system. Then when it is present in your blood vascular system, why not your aorta getting dilated? Why not your, let us say, femoral artery, abdominal aorta, carotid arteries, why are they not getting dilated? The reason is that low level of histamine cannot affect your blood vessels. Or in other way I can tell you is that blood vessels are not at all working, not at all listening, or not at all sensitive to low level of histamine. But what this pertussis toxin will do is that it will make that low concentration of histamine so important that all your blood vessels in your body will become so much sensitive to that low concentration of histamine. Okay, so let me draw some blue dots here. So these blue dots are representing your histamine. Okay, so these blue dots are representing your histamine. So how many histamine molecules are there? Very few. Still, even then, this pertussis toxin will make your entire blood vessel to become more sensitive to this histamine. So when your blood vessel becomes more sensitive to histamine, what is going to happen? Look here, lining your blood vessel, you have got endothelial cells. So these are your endothelial cells that are lining your blood vessel. All of you know that. So now when these blood vessels are becoming more and more sensitive to low concentrations of histamine because of this pertussis toxin, what will happen? Histamine causes vasodilation. Obviously, even this blood vessel which I have drawn here, it will vasodilate. So when it will vasodilate, what is going to happen, guys? The space between the blood vessels, or the space between the endothelial cells, the gap between the endothelial cells is increased. You see, I'm creating a gap between the endothelial cells. So as the blood vessel is stretched out, there are gaps left. Now what is going to happen? What is going to happen is that through these gaps, the fluid will go onto the surface of your cells in this way. You see, the fluid is going like this. Let me rub this cilia so that it will be clear for you. So you see how the fluid is going out. So this is how the fluid goes out, like this. And the fluid goes out, and there is a lot of fluid that is accumulated in the larynx lumen, right? A lot of fluid that is accumulated in the lumen here. Lumen of your oropharynx, lumen of your nasopharynx, you see. A lot of fluid has got accumulated here. So when a lot of fluid has got accumulated, don't you think the globulate cells will sit quiet? The globulate cells also will be activated. When the globulate cells will be activated, tell me what is going to happen? Yes, when the globulate cells will be activated, a lot of mucus is also released. You see a lot of fluid and a lot of mucus here. And compare the first cell to the second cell. This is the cell number 1, this is cell number 2. Even you find fluid between the cells. Intercellular, in between the cells also you find fluid. So overall what is happening is that this complete unit, or this complete pharynx, it is swollen up, right? It is swelling up. There is a lot of fluid that is coming from the blood vessel because of histamine. There is a lot of mucus that is secreted. So overall this entire larynx, whatever is there, it swells up. So look at the picture above now. Look at the picture here. What is happening here is that the entire thing swells up like this. You see how neatly you can see the swelling? So when this entire thing is swelling up, the lumen of the oropharynx becomes very narrow. So when the lumen of the oropharynx becomes very narrow, when the patient is trying to cough, obviously the cough sound has to pass from that narrow lumen. It will create a turbulence, and that is what is called as whooping cough. Okay. So now what is happening, guys? What is happening is, the second thing I have to mention is that, is that patient will have increased sensitization Increased sensitization to histamine. Okay. So what are you trying to tell us? What am I trying to tell you is that whenever there is increased sensitization to histamine, the patient will have laryngeal edema. Laryngeal edema. So whenever there is laryngeal edema, there is no surprise that obviously patient while talking or while coughing, he will have a distinct sound that is called as whooping cough. That is called as whooping cough. So you see finally whooping cough in the patient, right? So this is a particular whooping cough. Point number 3. Point number 3. Point number 1 was, what was point number 1? A subunit came inside the blood vessel and A subunit released pertussis toxin. First of all, pertussis toxin caused increased level of lymphocytes, lymphocytosis. Second, pertussis toxin caused the blood vessels to become more sensitive to histamine. Second, pertussis toxin is causing the blood vessels to become more sensitive to histamine. As a result, the blood vessels will dilate and all the fluid is rushed into the lumen, causing edema. So whenever there is edema, patient will have whooping cough. Third important thing is that, what is the third important thing is that, look here very carefully. Let me elongate this blood vessel, guys. Okay. So let me enlarge this blood vessel here. So let us say this is your blood vessel that is coming all the way down here, all the way down, and this blood vessel right now is supplying your most important organ, that is your pancreas. Okay. So this thing over here, this is your pancreas. All of you know that this is called as the head of the pancreas, this is called as a body of the pancreas, and this is called as a tail of the pancreas. Tail of the pancreas have got some very important cells like this. These cells are called as islets of Langerhans, right? So tail of the pancreas is having some specialized cells. These are called as islets of Langerhans. All of you must know, all of you must and should know what is the function of islets of Langerhans. Islets of Langerhans basically release a hormone that is called as insulin. So why are you telling all these things? We knew it. Then please tell me what is the difference between release of insulin from islets of Langerhans and you drawing this blood vessel all the way till your pancreas. What is the connection between both of them? The connection is that, look here very carefully, guys. I am drawing one islet of Langerhans cell here. Okay. So I am drawing one islet of Langerhans cell. So this is one islet of Langerhans. On the surface of islets of Langerhans, guys, on the surface of this, you have got a very special enzyme and that is called as adenyl cyclase. Can anyone tell what is the function of adenyl cyclase? You have studied in your pharmacology. Yes, adenyl cyclase will convert ATP all the way to cyclic adenosine monophosphate. And this cyclic adenosine monophosphate will in turn activate another group of enzymes that is called as protein kinase A. And what this protein kinase A will do is that, look here, within this islet of Langerhans there are vesicles. And what are these vesicles consisting of? The vesicles are loaded with insulin. So this protein kinase A will go and activate these vesicles too much that these vesicles will come all the way to the surface of the cell. Look here, the vesicle is coming all the way to the surface of the cell and finally the vesicle fuses to the surface of the cell and the insulin is released outside. So this is how normally the insulin is released. But insulin all the time cannot release in your body, right? Only when you eat the food, when you take glucose inside, that is a time you have to release the insulin. But the remaining time you have to stop releasing the insulin, because if you keep on releasing the insulin, that will end up having, the patient will end up having hypoglycemia. So how do you stop releasing the insulin all the time from your body, right? All the time you don't release insulin. So how do you stop the remaining time? How do I stop is that I have a very special protein that is called as G inhibitory protein. and this G inhibitory protein, time to time it will inhibit adenylyl cyclase. When it will inhibit adenylyl cyclase, the levels of cyclic AMP will drop, the levels of protein kinase A will be dropped, so obviously insulin cannot be released outside. But look here, the interesting thing. Within this blood vessel which I have drawn, you have got these structures, right? You have got these green color structures which I told you. What are these green colored dots? Yes, this green colored dot is nothing but your, yes, tell me, this green colored dot is nothing but your pertussis toxin. And this pertussis toxin comes all the way down in this way, and this pertussis toxin will enter into your pancreas, and this pertussis toxin, look here, this is inhibiting your G inhibitory protein. So when your G inhibitory protein is inhibited, will adenylyl cyclase be inactivated or activated? Obviously adenylyl cyclase cannot be inactivated, it will do its normal work. Normal work in the sense, it will convert into cyclic AMP, then protein kinase A, then insulin release. So can I tell, the third important thing is that pertussis toxin helps in increase release of insulin. Third important thing is that pertussis toxin will cause increase insulin release. So whenever there is increased insulin release, what is going to happen with this patient, guys? What is going to happen with this patient? This patient will have hypoglycemia. Hypoglycemia. The glucose levels are reduced in this patient's body. Clear? Right. So this is mechanism of one toxin which I told you. Okay, fine. So is that all? You told me three things here. One you told me is that lymphocytosis. The second one you told me is that this bacteria causes increased sensitization to histamine, that will cause laryngeal edema and whooping cough. And the third thing you told me is that it causes insulin release. Is there anything more that I can discuss? Can you discuss, please? The next important thing is that, guys, this not only releases this pertussis toxin, but it also releases one very important toxin. You know what will happen when the toxin is released? So this picture is little kind of messy, so let me draw a fresh picture over here very quick. So let me draw it here. So this is your cell, right? So this is your cells like this, right? And this is your blood vessel. And obviously all of you know that this is your lumen. Okay, this is your lumen. So right now in your blood vessel, what toxin is there here? There is A subunit. A subunit, previously it released one toxin that is called as pertussis toxin, and we discussed. Now this particular A subunit is releasing a second toxin. You know what this second toxin will do? This second toxin will constrict the blood vessel. It will cause vasoconstriction of this blood vessel. For example, this toxin is called as, let us say this is called as D, right? So this toxin name is D, for example. So this D toxin, what does it do is, it will constrict your blood vessel. What do I mean by constriction of the blood vessel? Look here now. So it is constricting, it is completely constricting your artery in this way. You see, it is constricting the artery. When it is vasoconstricting the artery, do you think there will be enough amount of blood that will be supplied to your cells, mucosal cells, I mean the laryngeal cells here, the pseudostratified epithelium? No. So when there is no blood supplied, then all these cells, they will die of necrosis. Understood? So they will die of necrosis. And this toxin is called as dermatonecrotic toxin. This toxin is called as, this D toxin which I told you, this is called as dermatonecrotic toxin. But anyways guys, if you wanted to remember the whole mechanism, which will be really useful for you to study the remaining subjects and the remaining parts. If you are perfect with the mechanisms, I am telling you MBBS is fun. So look here now. Can I list out all the different types of virulence factors or the toxins or whatever it is released? Yes. So let us list out one by one. Point number 1. Let us go back to the picture. The first thing happened, before this entire mechanism started, the first thing that happened is that this bacteria attached. This bacteria anchored to the host cell with the help of two things. One is pertactin and filamentous hemagglutinin. So, pertactin, second one is filamentous hemagglutinin, these are the two anchoring proteins. Next, third important protein or the toxin, whatever it is, what is it happening? It is releasing a tracheal toxin, right? So the third important thing is tracheal toxin. Next, the fourth important thing to understand, the fourth important thing is that the bacteria went inside, the A subunit went inside and it released what? Pertussis toxin. And finally, all of you know, the last toxin which is released is dermatonecrotic toxin. So if someone asks you what are the different types of toxins that are released by this bacteria, then one has to answer that these are the three different types of toxins that are released. So all these five constitute the virulence factors, but within these virulence factors, the two are responsible for anchoring and the remaining are the toxins. Okay, so all these which I discussed just now are your virulence factors of the bacteria. So I hope all of you understood the mechanism and I hope all of you understood why the patient is having whooping cough, right? It is all because of this pertussis toxin, you remember? So remember this thing, it is very important. Fine. Having said this thing, what will be the basic symptoms in the patient which I see? The symptoms of Bordetella pertussis in a patient, in a child, are classified into three. So what are these three? Okay. So what I was telling is the symptoms of this patient are basically classified into three important things. Now what are these three important things? The first stage is called as catarrhal stage. The first stage is called as catarrhal stage and this catarrhal stage will last for 1 week. This catarrhal stage will last for 1 week. What is the second stage name? The second stage name is called as paroxysmal stage. Paroxysmal stage, which is very important and it will last for about 1 to 6 weeks. Paroxysmal state will last for about 1 to 6 weeks. And the last stage is called as convalescent phase or convalescent stage. And what is this convalescent stage? This is the stage where the person recovers. This is the stage where the person will recover automatically by himself. So when the person will recover automatically by himself, so I think there is no need for me to use drugs in these patients, right? Only symptomatic therapy can be used. But after how many weeks it is happening? It is happening after 2 to 3 weeks. Okay. So can you please tell me that in catarrhal stage, what are the symptoms do you basically see? The symptoms which I see in catarrhal stage are patient will have low grade fever. Patient will have low grade fever and this is seen in stage number 1. Coming to the paroxysmal stage, guys, this stage is so severe that the patient will cough too much. There will be continuous cough. The cough will be so intense. So I am also writing it as intense cough. The patient will have intense cough. And obviously, what type of cough? All of you know that it will be whooping cough. Whooping cough. So patient will have very intense whooping cough. Now, because of this very intensified whooping cough, patient can sometimes vomit. And the cough is so intense that even sometimes the lungs will collapse, the ribs will break, you know. So, whooping cough is characterized in the paroxysmal state. The whooping cough is characterized by, first thing, patient might have vomiting. Second thing is that patient, let us say, he might have collapsed lung. Third thing is he might have broken ribs because of that severe cough. So broken ribs in the sense you can imagine how severe the cough would be. And fourth important thing is that as the patient is coughing, on the face there are small tiny capillaries, right? These capillaries, due to that high pressure, they will burst out. There will be hemorrhage of these capillaries. So when there is hemorrhage, you will find small red colored dots here. These are called as petechiae. You will even find petechiae in the patient, right? So these are the symptoms of the paroxysmal state. And in the convalescent state, what things basically happen is that the patient's symptoms start reducing. I mean, the main symptom here is the cough, right? So the cough gradually starts reducing down. Okay, fine. So I have divided these symptoms into 3 stages. Now can you please tell me how do I diagnose it? Should I diagnose looking at the symptoms? Obviously you can diagnose looking at the symptoms because they are so obvious, and the whooping cough will be so characteristic. You can very clearly, based upon the patient's presentation, you can very clearly identify the symptoms over here, the disease over here, what type of bacteria is affecting the patient. Apart from that, another important thing I want to tell is that, coming to the labs, what you basically do for any kind of upper respiratory tract infection, guys, let us say if it is COVID, what do? You do a swabbing, nasopharyngeal swabbing, right? So you basically do nasopharyngeal swabbing. Not only nasopharyngeal swabbing, after taking this nasopharyngeal swab, you put this swab, you grow this bacteria on a special agar that is called as Bordetella agar. That is called as Bordetella agar. Okay. Not only Bordetella agar, you can also grow it on Regan-Lowy medium. Regan-Lowy medium. Right. So how do I prevent this? How do I prevent this bacteria? Coming for the prevention, all have to know the important prevention point, that is you have to vaccinate the child. What type of vaccines can I give? I can give DTaP, right, or I can give TDAP. These are the 2 vaccines. Okay. So this is the prevention, the lab work, the symptoms, the overall virulence factors which we discussed, and also most importantly the mechanism, guys. So this is the pathogenesis and this is the mechanism which you have to definitely know. So overall, let us recap all of these things, guys. Coming to the pathogenesis, the bacteria enters into your nasopharynx. From the nasopharynx, it will reach the oropharynx. Once it reaches the oropharynx, with the help of anchoring filaments, it will, like filamentous hemagglutinin and pertactin, it will attach to the cilia. After attachment, it will start releasing a toxin called as tracheal cytotoxin. And this tracheal cytotoxin will paralyze all the cilia over there, so that the cilia don't move. Okay. Now this bacteria slowly, through a process called endocytosis, it enters. Basically this bacteria is having 2 subunits, one is called A subunit and B subunit. Together they form this bacteria. Okay. So this A subunit is released into the blood vascular system, and in the blood vascular system this A subunit releases a special toxin that is called as pertussis toxin. What are the functions of pertussis toxin? The first function is that it will increase your lymphocyte count. Second important thing is that it will cause histamine release, and it will cause the blood vessels to become more sensitive to histamine. So when the blood vessels dilate, all the fluid enters into the lumen, and this will cause laryngeal edema. So when there is laryngeal edema, when patient is trying to cough, right, what will happen? During cough, you will due to the turbulence, due to that narrow lumen, due to the turbulence, you will hear a characteristic oofing cough, right? The patient will cough like this. So he's producing a sound of oof, right? That is why it is called as oofing cough. Next important thing is that this toxin will even stimulate. It will even stimulate your islets of Langerhans. How does it stimulate? It will inhibit the G inhibitory. So when G inhibitor is inhibited, you know the entire process, and it will lead to insulin release, right? So when there will be insulin release, patient will have hypoglycemia. So this is the basic thing which you all have to know regarding Bordetella pertussis, and this will be very enough for your MBBS exams. Thank you so much for watching.